Welcome to the summary for Chapter 18 on proteins. Actually, Chapter 18. Things you must know from Chapter 18. Alright. Identify the characteristic parts of alpha amino acids. We have let me draw a couple amino acids here. It's an acid, so we have a carboxyl group, and it's an amino acid. So we have an amino group, and let's keep this simple side chain here. Okay, so the carboxyl group, because it's an acid, contains a carboxyl group, and at physiological pH it is ionized, so it will be the carboxylate, not the free acid, but the carboxylate with the ion here, -1 charge. The alpha carbon, you notice when I drew this, I started with the alpha carbon. That is kind of the centerpiece, the center of these amino acids. Of course, it's a carbon, so it's got 4 things attached. It's always going to have at least 1 hydrogen. It's got the acid group. It's got the amino group. So the amino group, put in blue here. The amino group there, NH3, also ionized, so it's actually an ammonium ion. And then the side chain, okay, side chain here. So we've got 4 things attached. Now there is one amino acid in which the side chain also is simply a hydrogen. That's glycine. Sometimes this is referred to as the R group. And there are 20 different amino acids. And they each have the same alpha carbon backbone, but the R group is different. So the R group, or the side chain, is different for all 20 amino acids. Something else you should be aware of, these amino acids are all chiral, well, except for glycine. Glycine has the side chain here as a hydrogen, so glycine, there would be 2 hydrogens attached to this alpha carbon, and it would not be chiral, but all the other amino acids are chiral. So where it says here alpha carbon, I'll say all but glycine, in which the R group actually equals a hydrogen, are chiral. And they are all the L enantiomer. They are all the L enantiomer. So in a standard Fisher projection, it would look something like this. And the carboxylate, here's the alpha carbon is chiral now, so we don't draw that. We have the amino group on the left, because it's L, hydrogen right, and then the R group, or the side chain on the bottom. So these are L amino acids, because this ammonium ion here, that amine, is on the left. The reason they're called alpha amino acids, again, is because let me draw another amino acid here. First off, I'm just going to draw an acid. Make it ionized, and this is actually valine. So the alpha carbon, this is carbon number 1. Carbon number 2 is the alpha carbon. Carbon number 3. The 3rd carbon would be beta. These are all alpha amino acids, because on this alpha carbon, carbon number 2, there's an amino group. And actually in all of these amino acids, except for 1, they are primary amines. So remember, this here is a primary amine. There is one amino acid, proline, which is a secondary amine. So let me draw the structure of proline here. And here's our alpha carbon, carbon number 2. I'll put the hydrogen down here. And the side chain actually wraps around and bonds, should be H2, bonds to that nitrogen. side chain here. Now it's going to wrap around and bond to that nitrogen. The nitrogen has now 2 carbon-carbon bonds, and so it is secondary. It's a secondary amine. The name of this amino acid is proline. It's the only one that is secondary. All others are primary. We also have one amino acid. These are all chiral. This alpha carbon is still chiral. As we go, here's the nitrogen, here's the carbon, here's the acid group. So it's chiral. It's got 4 things attached, all except glycine. Glycine is not chiral because the R group is just a hydrogen. So this alpha carbon has 2 hydrogens attached. Alright, so I'm not going to ask you to be able to draw or even identify or even name all the 20 different amino acids. There are some that you should know, of course, like glycine, which is a chiral, proline, which has a secondary amine, cysteine, which has a thiol group. But most of the others I'm not going to ask you to worry about. But I should expect you to be able to look at the amino acid, identify those different parts of the amino acid, and then classify these amino acids according to their R group or side chain. Okay. So the side chain, there's 4 different classifications. It can be nonpolar, polar, acidic, or basic. The nonpolar ones, these will just simply be, it could be just an alkane. So it could be a methyl group as an alanine. It could be an isopropyl group, such as you find in phenylalanine. It could be a benzene ring, like in phenylalanine or tyrosine. Could even have a thioether. So this is a thioether in methionine. Contains this thioether side chain, but most of them are simply, the nonpolar ones are alkyl groups. Okay, or they could be aromatic. Tryptophan is another one that has an aromatic side chain. Now the polar ones. The polars, these are either contain an alcohol, as in serine and threonine, or could actually contain, oh, it's an acid? No, this is an amide, as in asparagine and glutamine. So this is an amide. So we have those guys. We also have one that contains a thiol. This would be cysteine. I mentioned cysteine is important to know because it has this thiol and makes these disulfide bonds. We'll talk more about those in a moment. So cysteine, and you should recognize cysteine as containing this thiol. And we could have this amide group or an alcohol. Those are polar. Now they're neither acidic nor basic, so they're neutral, and polar neutral with regard to pH. So they're neutral and polar. Acid and basic, of course, they're going to be polar as well, but they contain an acid group. So we have glutamic acid, right. So the side chain also contains an acid. It says, let me go ahead and draw. Here's the carboxylate. Here's our alpha carbon, and then we could have one more. Glutamic acid. So there's an acid group also on the side chain. And the last one, glutamate and aspartate. These are the 2 acidic ones. Again, you don't have to memorize the names, but recognize the acid group certainly. And the reason I mentioned glutamate is because you may be familiar with MSG or monosodium glutamate. That's the monosodium salt of this amino acid. Actually, it should be NH3 plus charge. Okay. The basic ones, these guys contain amines. Kind of goes without saying, but let me, again, these guys contain a carboxyl carboxyl group or carboxylic acid. The amines, there's 3 of those. There's arginine, lysine, and histidine are the 3 that contain amines. I won't bother drawing the structures, but again, the side chain contains nitrogen. Not this nitrogen here, not the nitrogen attached directly to the alpha carbon, but the side chain here would contain a nitrogen. Right. And of course, at physiological pH would be ionized. Okay. Describe proteins in terms of the following characteristics. Globular proteins are spherical in shape. Typically they function as enzymes, or they could be carrier proteins, such as hemoglobin, we studied, or it could be an enzyme like hexokinase, maybe, more or less spherical in shape. For these globular proteins that are spherical in shape, they're kind of like micelles, so the nonpolar amino acids have their side chains on the interior of these globular proteins. Where it's hydrophobic. So the nonpolar hydrophobic side chains are on the interior, and of course the polar and acidic basic ones, the polar side chains on the outside where they interact with water, the outside interacting with H2O. So you'll see these nonpolar ones on the interior of a protein, the polar acidic basic ones on the exterior. And right, so there's they're also soluble, right? These guys are soluble. Important to remember. Contrast that to the fibrous proteins. Fibrous proteins are elongated or rod-shaped. Elongated or rod-like in shape. And these essentially are structural proteins, or maybe involved in mobility or motility, such as myosin. Myosin in your muscles. Myosin in your muscles. Or structural, we talked about collagen or keratin, elastin, these structural proteins. And of course, because they have a structural role, they're going to be insoluble. So actually, let me make a distinction here. Right. So this is in red, but the soluble here is referring to the globular proteins. The fibrous proteins are insoluble in water. Okay. Proteins are large polymers of amino acids, so they are greater than 50 amino acids long, whereas peptides or polypeptides, sometimes you'll hear referred to as simply peptides or polypeptides, these are less than 50, or less than or equal to 50 amino acids long. Proteins are greater than 50 amino acids. Peptides are less than or equal to 50 amino acids, and we'll explain why that is exactly down here in number 5, where we make this distinction. One thing I did not talk about in lecture was the fact that the average molecular weight, the average size of the amino acid is 110 grams per mole. Contrast this to water, which is 18 grams per mole. If you remember how to calculate atomic or molecular weights. And sucrose, chemical formula for sucrose, 340 grams per mole. Right? Well, if it's 110 grams per mole and it turns out an average size or typical size protein contains about 600 amino acids. This means that this thing is 60,000 grams per mole. So they're quite large and referred to as macromolecules. Draw the generic backbone structure of a polypeptide. So I'm going to put in maybe 3 or 4 amino acids, and again, I always like to start with the alpha carbon. I know the alpha carbon has got 4 things attached. I've got a hydrogen, I've got an acid group, and an amino group. So that's one. Now I'm just going to write this as a side chain. I'll make this R1 for side chain number 1. The next amino acid is in an amide bond. So here's the next amino acid. Here I've got the alpha carbon I just drew. The side chain is attached to the alpha carbon, so it's R2. Right, so there's the next one in purple. This carbon here is going to be attached to, say, amino acid number 3. Side chain, remember, always attached to this alpha carbon that has 4 different things attached. If I only have 3 here, then this is going to be the free carboxylate. So this, in any amino acid, or I should say in any polypeptide, there is a C terminus for the carboxy terminus, and the N end, or for nitrogen because it's an amino group, so the amino terminus, or the N end, amino terminus, and the C terminus. You read left to right, and they're typically drawn amino to carboxy, so A to C, also alphabetical. The peptide bonds are these amide bonds here. These are actually amide bonds, but when we're dealing with proteins, we refer to these as peptide bonds. Differentiate between amino acids, polypeptides, and proteins. Amino acids, remember, these are the 20 different monomers. 20 different monomers that go up to make peptides and proteins. And again, the big difference between these two is size. So polypeptides are smaller. Proteins, of course, are larger. It can be quite large. I'll set up here, typical amino acid might be 60,000, but it can go up to 6 million grams per mole, so quite large. And the reason we give them different names here, peptides versus proteins, is because if it's small like this, 50 amino acids or less, it doesn't really have a very well-defined secondary and tertiary structure that we'll talk about later. So these peptides are too small to have a well-defined secondary and tertiary structure, whereas most proteins, on the other hand, these are larger and they have a secondary structure. We refer to those, remember, as alpha helix and beta sheets, so they have secondary, tertiary, and even a quaternary structure. Know which stereoisomer of the 20 amino acids are used. Sugars are D, amino acids L. I mentioned that above. Okay. Know what is meant by amino terminus, or the N terminus, or N end, carboxy terminus, or C terminus. Remember, in a single amino acid, of course, you've got an amino end and an acid end, but when you string this together, you're only going to, in one polypeptide chain, you're going to have one amino end and one carboxy end. Know what a zwitterion is, an isoelectric point. Alright, so let's just use a simple amino acid, for example here, alanine. So here's our alpha carbon. I like to start with the alpha carbon. The side chain for alanine is simply a methyl group. Okay. Now I've got an acid group, and I have the amino group. Now in physiological pH, these are ionized. So physiological pH, generally these are ionized. When the pH equals the pi, the pi stands for isoelectric point. So pi, isoelectric point. Iso means the same. prefix iso means the same. What's the same? Well, the charge is, essentially this means the charge is balanced. We have the same number of positive charges as negative charges. So the net charge here is 0. Okay. If we raise the pH, high pH, right? How do we raise the pH? We add hydroxide ions. What happens is the hydroxide ion will react with this amino group and remove one of these hydrogens. So now the amino group is just NH2. Here's your alpha carbon. The carboxylate is still ionized, so now it has a net charge of -1. On the other end, if we lower the pH, we can do that by adding hydrogen ions. So when the pH is lower than the isoelectric point, again, the isoelectric point is simply defined as the pH at which this Zwitter ion exists. This is our Zwitter ion. Why is it Zwitter? Because zwei is 2 in German, and there's 2 ions, 1 positive, 1 negative. Net charge is 0. But at low pH, what we're going to do is we're going to stick this extra hydrogen here onto the carboxylate. So that carboxylate is now free acid. It's not ionized anymore. It's like that. Here's our alpha carbon again. And the amino group is still ionized, and so it will be NH3+. So now the net charge is +1 owing to this charge on the amino group. Okay. Primary, secondary, tertiary structure. Primary structure is simply the order of amino acids. Sometimes we'll refer to it as a primary sequence or primary structure. Order of amino acid sequence, generally written from the N terminus to the C terminus. Secondary structure, right? Actually, before I get onto the secondary structure, the primary structure, remember these amino acids are held together or bonded through these peptide bonds, held together by peptide bonds. Secondary structure is composed of 2 different structural elements. We talked about the alpha helix and the beta sheet or beta pleated sheet. Now the alpha here has got nothing to do with this alpha carbon. The alpha helix is simply the name because this is the first secondary structure that was identified, so it's given a prefix here, alpha. Beta pleated sheet was the second one identified, so beta. If we had a third one, it would be gamma, maybe. Now these secondary structures, these are held together by hydrogen bonds, and they are between atoms of the alpha carbon, get rid of atoms here, alpha carbon backbone. Okay, what is the alpha carbon backbone? The alpha carbon backbone is everything but the side chain. So here, these guys here, the amino group, the alpha carbon, this carboxylate, that's the alpha carbon backbone. So it's everything but the side chain. And the side chain down here. Tertiary structures? Well, once you get large enough, as I said, you can have secondary structures in a peptide, but once you get large enough, you can have 50 amino acids, you can have at least 2 different secondary structures, and these combine to form tertiary structures. So a tertiary structure is composed of 2 or more secondary structures. Polypeptides with fewer than 50 amino acids are generally not complex enough to have this type of interaction. Right? So they're just referred to as peptides or polypeptides. One important thing to remember here with these tertiary structures is the backbone, right? This amino acid alpha carbon backbone. This is what forms the peptide bonds for the primary structure, and also what forms the hydrogen bonds between the amino group of one amino acid and the carbonyl of another amino acid. So that takes care of the primary and secondary. The tertiary structures are held together by side chain interactions. So I'll say here these are stabilized by side chain interactions. Okay, so we have hydrogen bonds, we have disulfides, we have salt bridges, and we have hydrophobic interactions. Those are the main types of side chain interactions. All right, quaternary structure. A quaternary structure is found in what we call a complex protein. Right. So a complex protein has a quaternary structure and is composed of two or more separate polypeptide chains. That is, they have two or more primary structures. Right, so you'd have two N-terminuses, N-termini, and two C-termini. Each amino acid chain must have its own N-amino terminus and C-terminus. And these are held together by the same interactions as the tertiary structures, held together by side chain interactions. Examples of proteins with quaternary structures, we talked about hemoglobin. Right, hemoglobin was an alpha-2 beta-2 tetramer. I also showed you a structure of immunoglobin. Immunoglobin, specifically, it was IgG, and it was, again, we had actually, again, two different types of chains. We had two light chains and two, what we call two heavy chains. So with immunoglobins, they talk about light chains and heavy chains. I'm going to draw the structure real quick here for immunoglobin. So those are the two heavy chains. You kind of form this Y, and the light chains here. So again, it's an alpha-2 beta-2 dimer, or sorry, alpha-2 beta-2 tetramer. Describe the role of hydrogen bonding secondary structure of proteins side chain interactions. Okay, so there's all these different side chain interactions, right? Hydrogen bonds, these are going to be formed, you know what a hydrogen bond is. You have a hydrogen, and that's a through space interaction to maybe a nitrogen, or it could be also to an oxygen. Now the thing is, this hydrogen must also be bonded. Now this is the dotted line here is the hydrogen bond, right? But this hydrogen must be bonded to either a nitrogen or an oxygen as well. So remember, these bonds are polar, these are polar covalent bonds, and that puts a partial positive charge on those hydrogens. So the types of side chains that form hydrogen bonds, those are the type of functional groups we saw in the organic section of this course. Okay, so we have alcohols and we have amides. Let me show you the structure. So if we have a side chain that has an amide, looks like that. Salt bridges, this is going to be between an acidic side chain and a basic side chain. So the acidic side chain is the acid. Now it loses that hydrogen here, so it's going to be negatively charged. Where does that hydrogen go? Well, it goes on to the basic side chain. So instead of being NH2, it's going to be NH3, the plus charge. And again, we have this interaction between the positive and negative charge. Disulfides, this is that special one. Cysteine has a thiol. If we have two cysteines We'll put one in red, one in purple. Two cysteines come close to each other. These thiols react and we get a disulfide. And then lastly, hydrophobic interactions. Hydrophobic interactions, these come from the nonpolar hydrophobic side chains. So we could have maybe phenylalanine side chain with the benzene ring, or you could have a secondary butyl group as in leucine, or an isobutyl group as in isoleucine, something like that. But notice here, these are all there's no polarity here. There's no heteroatoms, no nitrogens, no oxygens, no sulfurs. It's just carbons and hydrogens. Describe the conditions that cause proteins to hydrolyze or become denatured and the results of this process. So hydrogen bonds are disrupted by heat. Also, we said agitation, just stirring it or mixing it or frothing it up. Salt bridges are disturbed by acid or base. Disulfides are disrupted by thiol reagents. So these are other compounds that likewise have a thiol on them, such as methane thiol. Hydrophobic interactions are disrupted by soaps or detergents. There's a whole lab talking about soaps and how soaps emulsify because they're amphipathic. They have a polar head group and a hydrophobic tail. Now notice, denaturation disrupts the secondary, right, that's the alpha helix beta sheet, and also the side chain interactions of the tertiary and quaternary structures, but it leaves the primary structure intact. Okay, so denaturation, we still have a polypeptide chain. It's not until we hydrolyze it, like boiling it in acid, where we disrupt the actually it's going to disrupt the secondary, tertiary, and quaternary structure as well, and that's because we're actually breaking the peptide bonds, so the primary structure as well. So hydrolysis involves breaking the peptide bonds that hold the individual amino acids together. So when you hydrolyze a protein, you release, hydrolysis releases free amino acids, AA for amino acid. Alright, so what are the ways, or what are the consequences of denaturation? Certainly loss of function, so let's say loss of structure, right? We disrupt the secondary, tertiary, and quaternary structures. Loss of function. Protein is not going to do what it's supposed to do. And also solubility. So precipitation. The protein comes out of solution. So it precipitates, forms a solid. What are some examples of ways in which your proteins could be denatured? So what I'm talking about here, your proteins, I'm saying, what might happen to you personally that would result in denaturation? Well, we said heat can disrupt those hydrogen bonds, right? So heat, we're talking about fever. If your blood pH gets out of whack, either acidosis or alkalosis, or alkalosis, this would be acidosis is low blood pH, alkalosis is high blood pH. Again, that disrupts those salt bridges, those electrostatic interactions. If you have, I don't know, maybe you're getting your hair done and you have a permanent or something like that, you could disrupt those disulfide bonds with thiol reagent. Hairdo or permanent. I can't really think of an example in which your proteins might encounter something that disrupts hydrophobic interactions. But you could also disrupt these thiols with heavy metal poisoning. You remember the cysteine, instead of being bonded to another cysteine, it could be bonded to a mercury or a lead atom, or I should say lead ion, mercury or lead ion, and instead of forming the proper disulfide, this will get cross-linked maybe to another protein. We'll indicate that with the red color, so it forms these cross-links and it precipitates, comes out of solution, destroys the function of the protein. So heavy metal poisoning forms these disulfide cross-links. Differentiate between lipoproteins, glycoproteins, phosphoproteins, being general term significance and function of each. Lipoproteins contain lipid. We talked about this in the last chapter. We had HDLs and LDLs, but we also have membrane proteins. So some proteins have a lipid attached to them as a kind of an anchor to the membrane. Glycoproteins contain carbohydrates or sugars. And again, a lot of membrane proteins are glycosylated or contain carbohydrates. I refer to these as cell surface receptors. We talked about this in the chapter on carbohydrates, the blood groups, for example. Blood groups A, B, and O contain different carbohydrates on their cell surface. Those carbohydrates, though, are not linked to the lipids, are actually linked to proteins that are embedded in that membrane. So these glycoproteins in the membrane have different sorts of carbohydrates on them, and that's what distinguishes blood type A from blood type B and so forth. Phosphoproteins contain phosphate ion. There's an old joke, there's two types of proteins in this world, phosphoproteins and proteins that have yet to be shown to be phosphoproteins. So many different types of proteins are phosphorylated. And one thing I didn't talk about this in class is the fact that phosphorylation of a protein kind of acts like a switch. So let's say we have a protein, let's say here, these are, call it a molecular switch. So you have a protein here, protein EIN, and we're going to add a phosphate to it. Now there's an enzyme that will phosphorylate it, and so now we have the protein with the phosphate actually attached to it, and there's an enzyme that will remove it. And often cases, when you phosphorylate the protein, it will turn the enzyme or the protein on. So here now it is active once it's phosphorylated. And if we need to deactivate that protein, we don't destroy the whole protein, just remove the phosphate, and now it is inactive. So phosphorylation like this kind of acts as a molecular switch, turning the protein in the active form and the inactive form. Oncogenes, proteins that are involved in cancer, are especially noted for this type of thing. Describe the general features of the secondary structural elements, alpha helix, beta sheet. It's referring to number 4 above, and really, again, here, this is where a picture is worth 1,000 words. So I'm not going to draw the picture. I'll refer you to the structure in the book, but recognize those structures. All right, lastly, a little bit more vocabulary. We talked about holoproteins, apoproteins, prosthetic groups. So we have, actually we also had pre-protein, pro-protein as well. So an apoprotein is inactive, but if we add the prosthetic group, it becomes active. This gives us the holoprotein. The example we used here was globin, those globin chains. Remember, we had two types. We had the alpha and beta globin chains. To that we will add the heme ring containing the iron ion as a cofactor. Use that term, cofactor. This heme ring is not protein, but is required for the protein to be functional. So the globin chains by themselves will not carry oxygen, but if we add the heme ring, which basically binds this iron, and it's the oxygen that binds the iron in the heme ring, very complicated, right? Then we get the holoprotein, we get the hemoglobin, which is an oxygen carrier, right? It carries oxygen from the lungs to the peripheral tissue. All right, so I think these are holoprotein, apoprotein, prosthetic group. Protein folding, just recognize that these proteins, they're not generally, a few exceptions, they're kind of wadded up or folded up into these spherical or ball type structures. Okay, if it is active and folded properly, we refer to that as a native state. If we do something, so this native state is active. If we do something, I don't know, maybe we heat it up or something, we denature it. It is no longer active. It is not the normal state, not the native state, is denatured state. All right, so there were a few other things that we talked about with proteins, but this covers quite a bit of what you'll need for the test on this next unit.